friends, welcome to another video tutorial from Shomu's Biology, and in this video lecture we want to talk about DNA supercoiling. Now, as the name suggests, supercoiling is really supercoiled concept because many people, I believe, they don't have a good grasp of supercoiling concept. So because there are different terms, different kinds of things written in different books and different pictures and diagrams. It's supercoiling can be best understood with the help of animation, because DNA supercoiling is a three-dimensional event. If you try to understand with a 2D picture, it will be really difficult. So always looking for animation for DNA supercoiling will be much easy. But I'll try to explain the theory behind the DNA supercoiling in this video. Hope you like the video and you probably understand the process. DNA supercoiling is the understanding, as you say, supercoiling. Why we call supercoiling? Why not coiling? The reason we call it supercoiling, not coiling, because DNA itself is coiled. So DNA contains two separate strands and both the strands are coiled one with another, spiral feature of the DNA. Okay, and as two strands wrapped around each other, that is itself coiling. So that's not supercoiling, that's coiling. So if I draw DNA structure, schematic drawing, and I'm not a good painter here, so I'll try to draw it my best. Let's say this is the DNA and this is the structure of the DNA. So if this is the structure of the DNA, then these are the major groups and these are the minor groups, right? And if you draw the structure of the DNA, both the strands, one is ranging from 5 prime, so this one 3 prime, another one is 3 prime to 5 prime. So this is the linear structure of the DNA, and this is spiral, that both the strands are wrapped around each other and bases are present in between. So this is coil form of the DNA, and DNA is coiled itself. So what is supercoiling? Let's assume this is a circular DNA. So let me draw the same thing for a circular DNA. So it will look something like this. Okay. And another one with a separate color if I draw. Okay, like this. Okay, so this is let's say a DNA which is covalently closed circular. So we call them CCC DNA, covalently closed circular. So this covalently closed circular DNA, as you can see, is a circle, but it's only a coiled form of the DNA. Now what is supercoiling? Supercoiling means the same DNA, if I draw it something like this, okay, something like this. So the same DNA, now let me change the course of how I am drawing it. Something like this. So another strand if I draw, will be like this one. So this point, this one, sorry, and this point, this one, this point, and okay, and this one, this one. So it will look something like this. I'm just trying to draw it arbitrarily because it's difficult to draw the picture here, so I'm trying to draw it. This is something like this. So this is a relaxed form of the DNA, although it's a closed circular DNA, closed covalently circular DNA, and this is not relaxed because this is coiled, only coiled. But now if you imagine the strands of the DNA is coiled on itself, so that is super coiled. That's the difference between coiled and supercoiled. That's why I draw this picture in front of you. I could have drawn that earlier before even start of the class, but that is the idea, although it's not exactly the same of between the structures. But remember one thing, so if you take one strand and coiling that complete DNA with on top of another DNA. That is supercoiling. So see here, once time that one complete DNA double strand crossing over another. So this is supercoil. It may happen twice, thrice, as many as times, okay? But that's the difference between coiling and supercoiling of the DNA. Now the question is, why this supercoiling? Because normally if you look at this structure, it's relaxed, and every situation molecules inside the cell always try to be in the relaxed state, because that's the most energy saving state. So the relaxed state is always preferable. So why this supercoiling? The answer to that is, let's say this is the relaxed DNA, no problem with it. Let's say it's a bacterial DNA, because bacterial DNA are covalently closed circular DNA. Now you want to initiate DNA replication. So bacteria want to initiate DNA replication. For that reason, it needs to separate strands from each other. So let's say this part of the strands are separated, and what they are doing at this point is they are starting the process of replication. So this part is open. So as this part is open, then what will happen? It is going to create tension to the rest of the DNA, a strain to the rest of the DNA. And to compensate that strain, the structure is coiled. That's why supercoiling is formed. And again, if you want to open more and more structures like that, it will form more and more supercoils like it. Okay, so let me explain this in another way. Let me draw this picture as it was earlier. It was like this. So it's through this and this. Okay. So if you look at the structure of the relaxed DNA, try to imagine one important thing here, that is the concept of linking number. Linking number. What is linking number, or L? Linking number is the number of times you need to cut one strand that will allow the separation of both the strands of the DNA. Is that clear? If you look at this picture, how many times you need to cleave one strand, which is, let's say, the blue one. Let's say the blue one. How many times, if you cut the blue strand, 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 times, you know now the blue and black will separate from each other. So the number of times you need to cleave for the separation of two strands from each other is known as the linking number. In this picture, which was 9. So 9 times you need to cleave. Now this linking number is always an integer value. It will remain an integer value. That means there is no decimal fraction of integer. It will be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, but never 1.2, 1.5, nothing like that. So this linking number for a closed covalently circular DNA will always be constant, unchanging. You cannot change the linking number if the DNA is coiled. That means somehow tied. There is no break. If the DNA is linear, then the linking number can easily vary. But if it's tied and closed-ended, you can never change the linking number. Now the constituent of this linking number is 2. So let me write it here. The constituent of this linking number is twist and writhe. So linking number equals to twist plus writhe. Now what are twists and writhe? Twist is the number of times one strand of the DNA cross another. So you may ask me, isn't this same as linking number? The answer is yes. It is same as the linking number if the DNA is present in a relaxed state. That means the DNA is present in a flat surface, in a relaxed state, right? But if supercoil is formed, then the twist and linking number will not remain the same. You know why? That's the term of writhe. Writhe is the number of positions or times one DNA complete two strands of the DNA crossing another two strand of the DNA. So this means 1. So the writhe of this structure is 1 in this case. Now if the structure forms something like this, let's say like this, so you can say the writhe is 2. right? And twist is completely separate depending upon how many times one strand crossing another. So if you look at this picture of a relaxed state of the DNA, what we can tell is the linking number equals to twist. In this case, twist, if you also calculate the number of times single strand crossing one another, is also 9. And the writhe, there is no writhe. So a structure without writhe means the DNA is not supercoiled. If it contains writhe, then only it means it's supercoiled. So now the writhe is 0. So the linking number and twist are equal for a relaxed state of the DNA which is present in flat surface. So now L equals 9. Now what will happen if we have one writhe? Because now imagine, as I told you, for a closed circular DNA, the linking number should be constant. Okay, keeping that in mind, as to keep the linking number in constant, we know the writhe is +1. Writhe is there. So the linking number should be 9. So to become linking number 9, where writhe is +1, the twist needs to be reduced to 8. Then we'll get 8 plus 1 equals 9. Try to understand this process clearly. The linking number should be constant, which is 9. To keep linking number constant, if writhe is 1, then the twist should be reduced. Right? And there are also two types of this writhe, positive and negative. So the value for writhe can be plus or minus. In this case, we see it's a positive writhe. The value was +1. That's why the twist is decreased. If the writhe was a -1, the twist should increase. So if the writhe is -1, the twist should be 10 to compensate the value of linking number. That's the mathematical aspect of the structure. Now why it's happening? Try to understand one thing very clearly, is that DNA, which is a normal common form of DNA that we know, is right-handed helix by its own. On its own, it's a right-handed helix. So if a right-handed helix is having a right-handed supercoil, right-handed coil means clockwise, the rotation of the DNA onto another is clockwise. That is known as the right-handed helix. Okay, so the right-handed or clockwise turn, if you imagine here, for a right-handed, so a right-handed DNA having a right-handed turn, what it will do? It will cause a positive writhe +1. So if you have a positive writhe, the twist value should decrease to compensate and accommodate that writhe. Otherwise, the structure will be unstable and DNA will break apart. The same thing, as the DNA is right-handed, if it forms a left-handed writhe, that is counterclockwise writhe. So for a left-handed writhe, what will happen? The value of writhe is minus. So to compensate that, the twist number should be increased. So if the writhe value in this case is -1, so to keep the linking number 9, the twist should be increased to 10. That is, twist is increased by +1. That is the idea. So whenever twist is somehow introduced, the writhe should be in negative. Twist is reduced, the writhe should be in positive value. Now a very important question, because during the process of cellular events like replication, transcription, the DNA strands need to be unwound and separated from each other. So once the DNA strands need to be separated from each other, at that position the twist number always alters, right? We are altering twist number because we are separating strands. So as we are altering twist number, how can the linking number be constant? Because if we change the twist to accommodate and hold the same linking number, writhe needs to be added. It's a positive writhe or negative writhe. For example, let me show you one situation here. During the process of replication, we are opening the strands from each other. So let's say the linking number was 9, twist plus writhe. So what we have done here, we've decreased the twist. Let's say earlier the twist was 9 and the writhe was 0. So now we somehow decrease the twist to 6. So to compensate the value of twist, what we need to do? The writhe, there should be 3 positive writhe. So 3 positive writhe means the DNA will be supercoiled. Like That means there will be 3 separate positions when the DNA is crossing each other, right? But that is another problem, because let's imagine this is a portion of the DNA, we are separating the strands of the DNA, and if that causes positive writhe somewhere here in the DNA, it creates knot in the DNA downstream. And let's say this is the polymerase is moving in this direction from 5 prime to 3 prime, and as if you look at here towards 3 prime, that is the downstream of the polymerase movement, there is formation of supercoil positive here. Formation of supercoil formed to the downstream, positive writhe value. So how to prevent it? We need to change the writhe. We need to cut and remove the writhe so that the replication or transcription, whatever machinery that we are going, should continue. So now we are artificially from outside changing the writhe, but twist number is not changing. Twist number is the same. We are changing the writhe to 0. So what's happening, literally the value of linking number is becoming less compared to the one that we started with. I told you always that the linking number is an integer and that is unchanged, but during this physiological process, the cellular processes, the linking number is altered, and it needs energy to alter the linking number. It needs energy to resolve these supercoils. And who does that? The enzymes known as topoisomerase. So topoisomerase enzymes like topoisomerase type 1 or topoisomerase type 2, it can alter this writhe value. Topoisomerase 1 changes the writhe by 1. So it can cleave 1 DNA strands and can release, and what it can do, it simply opens up one twist that can also alter the writhe value. On the other hand, topoisomerase type 2 can clip both the strands of the DNA and pass one strand from each other. That changes the writhe by the value of 2, right? Now as we are introducing this topoisomerase which is changing our writhe, that our DNA is no longer supercoiled so that the process of transcription and replication can continue, that also altering the linking number. So the linking number that is changed now is the actual linking number, and the linking number that we've started with, which is theoretically should be 9, is known as linking number naught or LK naught. The linking number where there is no writhe, that is 9, known as linking number 0 or L naught, and this one is L or the actual linking number. Then what we need to do is we need to find out the difference between this, so del L linking number naught minus the linking number, actual linking number. So if we find between these two linking numbers, actually L minus L naught, that's the difference we want to find. Now if we find here that the difference value, let's say in this case L is 6, L naught is 9, so the linking number difference is minus 3, which is less than 0. So if this linking difference is the linking difference between the linking number at the relaxed state and the linking number during the physiological process. So if this del L value is less than 0, we call that supercoiling as a negative supercoiling. If the del L value is greater than 0, let's say plus 2 or whatever greater than 0, we call it positive supercoiling. Okay, so for a positive supercoil to occur, what we know is that it will change the linking number, and also negative supercoil change the linking number, because we are introducing different proteins and enzymes to do that in place. That is the idea. So remember one thing very clearly, that the positive supercoiling means on the left handed turn, and that is a clockwise direction, while a negative supercoiling, right handed turn, anti-clockwise direction. That's how the supercoiling is explained. So these are the mathematical components of supercoiling, and try to understand that normally in our cell most of the DNA is present in negatively supercoiled state. You know why? Because negatively supercoiled state of the DNA is very important, because it's kind of compensate when the DNA strands start separating, because of DNA is earlier negatively supercoiled. Even though the DNA start melting and strands start separated, even though we increase the writhe values, it will still reach the actual relaxed state drinking number. So it actually help in compensating the effect of addition of writhe. That's why normally DNA is present in minus or negative supercoiling state. But positive supercoils formed when you have continuously separating the strands, and after passing several twists to be separated from the two DNAs from each other, it can cause a positive supercoil downstream of it, which needs to be removed with topoisomerases. That is the idea of DNA supercoiling. And this is also an idea regarding DNA topology, which is regulated with the topoisomerase 1 and topoisomerase 2 enzymes. And remember, both topoisomerase 1 doesn't require any energy, but topoisomerase 2 requires ATP for the process of changing the topology of the DNA. Okay, so that's all about DNA supercoiling. I hope you understand DNA supercoiling. I'll try to make an animation of DNA supercoiling which may give you better understanding in three-dimensional space, but I hope I can explain the idea of topoisomerase and the role of supercoiling in the DNA functionality in the cell well in this video. If you like this video, please hit the like button, share this video with your friends, and definitely subscribe to my channel to get more and more complex topic videos like that. Thank you.